Let's go. There it goes. Hi, I am Miss Sloan, and this is APBio, and we are reviewing Chapter 17. Oh, hello. Chapter 17, Speciation and Macroevolution. Here are the big ticket items that you can read through. Just take a quick minute and skim them, please. Hi, welcome. Come on in, guys. Here's the code real quick. Like, memorize it. See if I can get it back out there for you. Maybe not. It is ZS. Okay, there it is. ZSUUWJ. Hurry, hurry, hurry. You're late. Because of my song. ZSUUWJ. The rest of you can still see the slides, so you can keep reading, right? Yeah. Okay, good. By the way, I threw down a really good TikTok you guys should be checking out. It's all about chapter 18. It's very good. I'll put it on Instagram. You don't have to get TikTok. Okay, does everybody have who wants? Got the code? All right, I'm going to keep going. All right, so let's go through. So on the species, you're chatty. On the species definitions, remember I gave you these kind of as comparisons and for us to fall back to occasionally when we have trouble with the biologic species definition. So when we look at morphological species concepts, who does that work really well for? Morphological. Fossils, right? Fossils, because we don't know behavior, we don't have access to DNA, right? We don't know any of that. So it is good for fossils. And if you are asexual, then we looked at the phylogenetic and evolutionary species. Those are basically one and the same, where you're looking back at your ancestry. But at what point do you declare it a species? Where do you draw the line if you're just looking at that phylogeny? When do you say it's a new species? So that's a problem as well. So the definition that we use is the biological species concept. And basically it says that you're a population that does what? Yeah, you interbreed with each other, and it's not successful for you to interbreed with anybody else, right? And the problem is we don't know when you're separated allopatrically. You know what that word means. When you're separated, maybe you would interbreed, you're just not near each other. Or maybe you interbreed in a lab and you wouldn't do it in nature, or vice versa. So, and then for those organisms that are asexual, then that becomes a real problem. So that's when you're probably going to go with the morphology. Okay, so that was the big ticket items on species. Okay, then number 2 were reproductive isolating mechanisms. So these can be structural, functional, or what? What's the next one? Behavioral characteristics that prevent successful reproduction from occurring. This can cause you to diverge from one another and then maintain your speciation. It's like, why aren't they interbreeding? Why are they considered a separate species? Because they have these reproductive isolating mechanisms. Yes?
What are the differences between evolutionary and phylogenetic?
I said there wasn't much. That's what I just got.
Is there any difference?
Not much. Not for us. We don't have to worry about it. Oh. Yeah, that's what I was just saying. Okay, so then I did the summary of the reproductive isolating mechanisms, so we can go over them. So geographical, big, allopatrically separated. So obviously you're not interbreeding. Ecological, what one is this one like? Habitat, right? We are living in the same area technically, but I'm swimming in the middle of the pond near the surface and you're swimming at the bottom. I'm on the edge of the tree and you're to the interior of the tree. So we've kind of done some resource partitioning in our habitat, and so that's why we don't interbreed. Temporal isolation has to do with time. It could be with big things here, like what are you seeing here? Differences in seasons, but it could literally be hours in the day. We looked at some, I think it was fruit flies that wanted to interbreed in the morning and others wanted to interbreed in the evening. And I would say if you're preparing for this quiz, you might as well prepare for your unit exam. And I need you to know all their names and have an actual example that you could explain to me. And do you remember how I embedded in the presentation? Like besides the presentation ones I gave you, but there were ones like that website that went through all those different examples, and just have a working one for each. Okay. Then we talked about behavioral isolation was next. And on behavior, it's how you communicate, right? That takes us back to communication, how you indicate that you're ready to have sex or not have sex. So that can get in the way. It might be, especially with birds. We looked at all those examples like the bowerbird and the song and dance that you have to do in order to indicate that you want to have sex. And then remember the jumping spider one? That was good, right? So what did he have to do to try to not get eaten? Dance and jump, right? That's what he did. Did he ultimately end up getting eaten? Yes. But did he pass on his DNA? Yes. So is he still fit? Yes. Okay. Mechanical isolation, your parts do not fit together. And the really good example we did for that one was the dragonfly and the clasper. Remember the species that the male cannot hold a female of a different species. Gamete fusion. So it could be that you get all the way to the point where the sperm and egg are right next to each other, but the female does not have the right receptors for that male. Or the example we looked at with plants, where when the pollen and the sperm nucleus and the tube nucleus are growing down, that they're blocked. It would be like a diaphragm blocking the sperm from ever meeting the egg, if you know what that is. Sorry, just threw that out there. Yes.
So an example of gamete fusion would be the receptors not meeting, or would it be the plants?
I gave you two.
Okay, so both of them are examples.
Yeah, both of them are examples. Okay, then let's say they actually do fuse. So these are talking about post-zygotic. So you're actually making a zygote. So sometimes because the chromosomes are not homologous, because they're not the same species, you have no homologous pairs, development is interrupted, and you have a miscarriage or a spontaneous abortion, or it never actually develops. Okay. Then, so that would be hybrid inviability, right? It's not able to survive. Sometimes the offspring is formed, but it doesn't ever reach maturity, right? It cannot reach maturity in order to reproduce. Or maybe the hybrid is sterile. Or maybe the hybrid isn't sterile, but the F2, right? The hybrid would be F1, the first generation. Maybe the F2 is sterile. So keep in mind, we're working from farthest out to the end, right? And you don't want to waste your sperm, egg, or your dances for somebody who's not going to increase your fitness. So the earlier the interruption, the better. When you get down to post-zygotic, that's a complete waste, right? Because you've given time, energy, nutrients in order for that young to develop. All right. Third topic, okay, was modes of speciation. And so I just wanted to throw them all on here. We talked about allopatric versus sympatric, right? So allopatric always starts with a barrier. And then if you actually get speciation, then when you're reunited, when the barrier is removed, that's when you know. You got to have the barrier removed to know if the differences have accrued, right? You're going to have different initial, did everybody sign the sign-in? You're going to have different initial gene frequencies. You're going to have different selection pressures. You're going to have different mutations. You're going to have different habitats. All of those things, you can accrue differences. And if you become sympatric, which means you're right there next to me now, and you have lost your ability to interbreed, then speciation has occurred. Now, sympatric speciation, you were right next to me, right? And there's two big ways that you get sympatric speciation. Little microhabitat changes, right? Like maybe the habitat itself or the climate changes. One goes towards the shade and one goes towards the sun, or maybe trees start to grow. So one starts to live in the trees and one starts to live on the ground. So you have little microhabitat changes, even though we're right there in the same habitat wanting to have sex at the same time. And that can lead to speciation, or it could be what? What's the other one? Chromosomal. And what were the two chromosomal ones? Autopolyploidy and perfect. Okay. And that was the two sympatric ones. Okay. Then we have things like divergent evolution. Divergent evolution is where we started the same, right? And you can do a little bit of divergent evolution and roll it on over to adaptive radiation too, right? But that you have speciation because one is focusing on, something slightly different, or the environment changes, or they're traveling to new location and they need different adaptations. You might see that in divergent evolution. Adaptive radiation, all of a sudden there are vacancies and you're going to exploit them in order to reduce competition, right? So like when the dinosaurs became extinct, you had this mammalian explosion that would be, radiating out. We always look at the example of Darwin's finches or the honey creepers, Hawaiian honey creepers, where they just become so competitive that I'm going to just have a little bit smaller seed, and it's about specializing, right? It's becoming specialist to a particular area of that habitat in order to reduce your competition. So these are all modes of speciation, splitting one species into two or more. So then I did have, for allopatric, I had a slide just for that, but I also wanted to talk, what do we call this species like this when this happens? Ring species, good. And then we talked about this, like you were all on the same island and you were competing for the same resources. Maybe a storm happens and some finches get blown to a different island, where if you're going to survive, you got to eat what's available, and then you become specialized to that. So if for some reason you go back onto the original island, you have already accrued differences and can exploit that difference on that island. Yes?
Does allopatric speciation lead to sympatric speciation? Or
No. Allopatric and sympatric are two different things, but what I want you to be familiar with is the words, because allopatric speciation starts with a barrier, so that when you are side by side again, and that's called you're living sympatrically, you're a sympatric, you're living side by side, you are different. Sympatric speciation is we are sympatric, that's how we live, and we still changed. Okay. All right. Then when we go to sympatric speciation, there is not a geographical barrier. A mountain, a river did not separate you. You lived right side by side. There could be slight little microhabitat changes in behavior. It could be a mutation. Maybe you have a mutation and you can't lift your wing as high, so that when you communicate that you're ready to reproduce, this is as high as you go, and maybe some others have the same mutation, and then you just start interbreeding with each other, right? It could be, but you were sitting side by side. Now, on polyploidy, this is more likely. For sympatric speciation, it's more likely to be a chromosomal issue. This would be less likely, okay? For polyploidy, what does poly mean? Many. So we're dealing with extra chromosomes, both scenarios, okay? Scenario one, in autopolyploidy, you are one species who's given rise to a different species, okay? One species given rise to a different one, probably because of failure for chromosomes to separate when you made your gametes, non-disjunction occurred. So all of your gametes, instead of being they are what would you say? Instead of being, what are your gametes supposed to be in relative to you? Haploid. But let's say instead you make some diploid gametes. Perfect. If those two diploid gametes, like, because if you can self-fertilize, right? If those two diploid gametes fuse, then you can form a then 4N zygote. This is more likely to happen in plants than animals, right? Because we just get one wrong chromosome or an inversion, we have trouble, right? So this is not typical of any animal speciation. Animal speciation, it's going to be all about allopatric speciation. It's plants that could have, right, the sympatric speciation where you have the polyploidy, okay? Now, allopolyploidy, two different species forming a third species. And in this case, you each produced your gametes, and your gametes fuse, but are they homologous? No. So you would think they would break down, right? But instead, they do a quick mitotic event. So now you have two of every chromosome. So technically you have homologous pairs. Those homologous pairs are going to be exactly the same, right? Because they came from the same chromosome. But now you can do meiosis, produce gametes, and continue forth. Okay? The way I remember allopolyploidy is there's two L's and I think two different species. Autopolyploidy is just one species becoming another. Questions? Yes?
So if there's a barrier between two species, would you say that they're living allopatrically?
Yes.
So, but you don't have speciation, okay? So,
You won't know. I mean, you can look and observe and see who else they mate with, but you really know once those species have reunited. Now, obviously, if you see they've accrued differences and they never reunite, yeah, you could still say speciation occurred. But it always starts with a barrier. Okay, what else? All right. And then let's revisit these ideas, right? So we talked about homologous and analogous structures, and it's important to talk about this when you talk about speciation, because you're not going to say, oh, we're the same species because we have wings, because wings could be a result of what kind of convergent evolution. So you need to look at the structure, the anatomy, and that's when you're relying on the morphology, right? That's when you're looking at the anatomy to decide, hey, how is this formed? But you can back that up because we know any morphological characteristics that we have. Why do you have those characteristics? Because what created that? The DNA. The DNA coded for that anatomy, right? And so you want to make sure that when you're looking at your species that you differentiate between analogous structures, which are purely talking about function. Now, of these that are here, these all have wings, so they are all analogous, but some of them are what? Homologous. The bird and the bat subtopic, they are homologous. They have those homologies in their structure, so probably they have the homologies in their DNA. Okay, and then just going over divergent, parallel, and convergent evolution. So remember, convergent, we talked about we are not related to each other, but we ended up on the same place, and those are typically called what kind of structures when you do that? Starts with a name. Yeah, analogous structures. Divergent, we had a common ancestor, wolf. Okay, now we have all these species of dog. Okay, now can dogs sometimes interbreed and you get some weird mutt things? Yes. So that's when you look at interbreeding and going, they're just doing it because they can, but we still call them two different species of dog, but we have the bigger, like those are subspecies, right? We still, they're all called dogs, even though they're interbreeding, but you have subspecies of that. Parallel evolution, we had common ancestry and we ended up in the same spot, which would be like a convergence, right? But we traveled down the same pathways to get there. So that's why I say in here that parallelism and convergence are not clearly distinguishable. So if they're not, I wouldn't worry about that. I know what they will address are things like analogous and homologous, right? And that you can look at homologies in DNA, you can look at homologies in like genes and those kind of things. Yes?
Would divergent evolution be both allopatric and sympatric or just allopatric?
Depends on how it formed. Divergent just means we had a common ancestor and we went our own ways. Yeah, you could do it allopatrically or sympatrically. Okay, what else? All right, next. So the principles of macroevolution, there were a couple things here. One was, I need to move myself here. Can I move myself over here? Keep going, Winnie. Go. Go. Okay, you can see it now anyway. Hopefully I won't be in the way for the next one. We talked about the pace of speciation, whether it was phyletic, that's like looking at phylogenies, where you came from, phyletic gradualism, or punctuated equilibrium. Those are just the pace of how quickly it happens. Phyletic gradualism is much more, what is it? It's very slow, and you're going to see more transitional fossils. Yeah. Punctuated equilibrium is very quick. By quick, we mean speciation occurs in about 1 million years. Okay. And you're going to see fewer transitional fossils. Okay. That's the pacing. Okay. But it's also about genes and when you turn them on, when you turn them off, and how long. Right. So, oh, great. I'm about. So we talked about heterocrony, right? And this is the timing and development of events. We talked about how you can get speciation. We have the same genes, right? We all have phalanges, but a bat has their phalange genes turned on a lot longer than we have our phalange genes turned on. So that's what that's talking about. You can still get new species. Huge things. We can't fly with our fingers, right? So, but bats can. So, big difference in characteristics, but same gene. Just how long the switch is on is what has varied. Okay, and okay, then we talked about those developmental switches, and we talked about similarities, and we talked about Hox genes as well, right, that are coding for transcription factors. And then I think I put a bunch of them on one page. I didn't want you to think you needed to memorize them. You don't. I just want you to see, like, there is a gene that codes for eyes, and these are totally different types of eyes, but it's just that same gene and modification in the switches. There's a gene for limbs and birds and animals. Same gene, just timing. We talked about Hox genes, whether you're a fruit fly or a mouse or a human, similarities there. And then the PIDX1 gene just had to do with what promoters were available or not, depending on how that gene was expressed. So it's not like we have to come up with all these different kinds of DNA. It's just timing and switches. And then we talked about what is and what, oh, look, I'm an 8. What is and what isn't evolution. And we remember it's not goal-oriented. Humans are not the end result, right? There are branches that die, branches that are successful. Mutations can bring about variation. Natural selection acts on the best genes and promotes those, right? And keeps them and conserves those. Are we perfect? No, we have a lot of inconsistencies because there are a lot of trade-offs that are made. Like, it's more important to be upright than to have a good back, right? So those are all those kind of contradictions. And then that was it. Y'all have any questions? I don't think the quiz will be hard because I haven't written it yet. Usually I haven't written by this time. Okay.